Jane Figueiredo from USC Keck School of Medicine in Los Angeles. She's an expert on the molecular and genetic epidemiology of cancer. Her research focuses on applying epidemiologic methods to investigate how genetic and environmental factors like nutrition or diet contribute to the onset of various cancers, including colorectal cancer. The title of today's talk is Genome-Wide Studies of Gene-Dietary Interactions and Colorectal Cancer Risk. Jane, please.
Well, thank you for the kind introduction. It's wonderful to be back in Germany. And thank you especially to the organizers for a fantastic dinner yesterday. So it's, yes. Well, especially for the dinner, and today's talk will talk about gene-dietary interactions. So just to begin, yesterday I think several of the speakers had talked about colorectal cancer. There's over 1 million diagnoses of colorectal cancer throughout the world each year, affecting about 1 in 20 males, a little bit higher than for females. And dietary factors are known to be important in cancer risk and mortality. And this was hypothesized quite a long time ago by Dahl and Petel, very famous epidemiologists, that estimated about 35% of cancer deaths were attributable to diet. And this really ranges depending on what type of cancer, between 10 and about 70%. And for colorectal cancer specifically, there have been a number of noted important factors, including red meat, processed meat, fruit fiber, vegetables, and folate. So for red and processed meat, there is convincing, it's now considered convincing evidence, according to the World Cancer Research Fund 2007. But a lot of the studies, there's been tremendous amount of variability in most of these observational studies. And some of this has to do with differences in gender and by site, whether we're talking about the colon or the rectum. But overall, our estimate for the relative risk is around 1.5 for red meat, and it's a little bit lower for processed meat. And what I mean by processed meat, I'm going to spend some time talking about it, is any type of food that's been packaged, cured, or processed. In countries like Canada, it's mostly cold cuts and bacon. In other parts of Europe, it could be also sausages and those types of food. And the estimate for that is slightly lower. And most of the hypothesized mechanisms have to do with the processing or the preparation of meat that generate different carcinogens, including heterocyclic amines and polycyclic aromatic hydrocarbons. And in processed meat, we often suspect that it's nitrates. So fruits, vegetables, and fiber were at one point considered convincing evidence that they protect against colorectal cancer and potentially other cancers, but it's now been downgraded to probable or limited suggestive evidence. And this is because there's been a lot of inconsistent observational studies and meta-analysis. In particular, from the Nurses' Health Study, that was published in the New England Journal of Medicine in 1999, they didn't show a protective effect for fiber. The European study from EPIC did in fact show a protective effect, and so there's been a lot of discussion about whether or not that is actually true. And for fruits and vegetables, the evidence is probably a little bit more inconsistent across studies. For folate, folate and folic acid, this has been a huge area of research in colorectal cancer, but it remains very controversial, with a lot of studies showing either a protective effect or actually an adverse effect. And we obtain folate from different sources, from either our diet, which is the only source in Europe, but in countries that have fortified their food supply with folic acid, that includes the US, Canada, and Australia. This is in the form of folic acid, which is the synthetic form of folate, and we also obtain it in multivitamins. And for this evidence, it's been, from observational studies, there's been a beneficial effect of folate that has come from many of the case control and cohort studies. But from the trials, I believe that you talked about the polyp prevention study yesterday in terms of aspirin, but we also looked at the role of folic acid in that trial, and the trial showed an adverse effect for folic acid promoting growth of adenomas, advanced adenomas, and multiple adenomas. And so this is because folate plays a critical role in nucleotide synthesis and in methylation reactions. And certainly what we understand right now is that deficiencies in folate are implicated in many disease processes, including colorectal cancer. But at some point of sufficiency, there is unlikely to be a beneficial effect, in particular among individuals that have pre-existing lesions. And so in terms of the genetics of colorectal cancer, colorectal cancer is a heritable disease. We estimate it's around 35%, which is probably a bit lower than some of the other cancers that we suspect. And what's been interesting in colorectal cancer is that this part, there's been a number of genes that we have identified mutations in, and these represent some of the hereditary forms of colorectal cancer, including FAP, Lynch syndrome. And initially we thought that this proportion would be a lot higher than it really is, but it's about 5%. So it's 5% of the 35% that we suspect is contributing to colorectal cancer. And so this led open the hypothesis that there were a number of common variants that were more frequent in the population, except that they didn't have a very high effect. Unlike these rare mutations, which confer a high risk of colorectal cancer for those individuals that actually have that. So I'm going to spend most of my time talking about this area of the research, but I wanted to note that right now we've also been trying to look at variants in this region through sequencing efforts. And so the genome-wide association studies became very popular, and that made this cover of Science. We call them GWAS studies, and essentially they are case control studies or nested case control studies where we profile individuals in a control population and in a disease population, in this case colorectal cancer, and these are genotyped on different platforms. The first study in colorectal cancer was only done on a platform of around 100,000 genetic changes, and then we plot them, and so this is just a plot of a Manhattan plot of the chromosomes where we've identified different genetic variants at a high p-value. And so just so everyone's familiar with what a SNP is, these are common changes in our genome where some of us will have a C allele and other may have a T allele. They are not necessarily pathogenic, but they differ between people at a frequency about 1%, and they make us all unique, and we believe that they contribute to susceptibility to different diseases. And there are many SNPs that we are studying will be genotyped, but what we can do is that because they are in high LD or they're correlated to many other SNPs, we can actually impute these genotypes. And so for colorectal cancer, there's now been several GWAS studies that have been conducted, and what we know so far is that there are around 30 markers throughout the genome that occur on actually these chromosomes that I've identified here in red that are related to the risk of colorectal cancer. But what's interesting is this now only comprises about 8% of the 35% that we now think is important is the hereditary component in colorectal cancer. So that still leaves open a huge amount of what we call the missing heritability factor. There are 2 publications that I know of that have been submitted, so we have at least another 10 GWAS loci, but they're unlikely to really capture all of the hereditary that we believe is going on. And so one possibility is that there are, at least in colorectal cancer, a number of gene-dietary, which I'll focus on, or other types of environmental factors that may be important and may be explaining part of this risk. So today I'll just focus on red meat, processed meat, fruit, vegetable, fiber, and I'll talk a little bit about some of our results with folate as well. But so what do I mean about gene-environment interaction? What I mean is that for individuals, so in this, if the E in this case is red meat, we do believe that if we consume higher levels of red meat, we'll have a higher risk than individuals that don't consume red meat. But there may be genetic factors that may make some individuals have a higher risk of when they consume red meat compared to other individuals. And this is what we're examining. We're trying to identify genetic changes that differ in the population that change the effect of meat. And this is particularly important because this may help us understand the etiology of colorectal cancer, but also it's important in terms of prevention or targeted prevention strategies, which I'll talk about a little bit later at the end. And so our first attempt to do this was actually done, it was published in 2011 in CVP, and it was done using the Colon Cancer Family Registry in the United States and in Canada, where we looked at 1,000 cases of colorectal cancer and 1,000 controls, and we identified out of 1 million genetic changes, only one gene dietary interaction, which was not very significant. And so we tried to replicate it in the Arctic study, which was done in Ontario in Canada. And as you can see here, we were unable to replicate these results. And of course, part of the issue here with these types of agnostic searches of looking at the genome and interactions with diet is power. And so with 1,000 cases and controls, we had done these calculations and essentially showed that we had very little power to detect what we would have estimated to be an interaction, an odds ratio with an interaction that was more reasonable. When in fact that we needed was about 10,000 cases and 10,000 controls in order to do these types of analyses. And so there have been a large number of consortium that have been formed that have been dedicated to try to do these analyses in a much better way. And so the work that I'll talk about here today comes out of the Genetics and Epidemiology of Colorectal Cancer Consortium, which really is the efforts of many of you sitting here who have participated in this research. Some of the largest studies include the German study DOCS with a large number of cases in control. Also from Canada, Australia is also on this list, and the rest come from the US. What's important here is that there are about 20,000 cases and over 20,000 controls. These have been genotyped on a variety of platforms, but they also have harmonized dietary and genetic and environmental data. So because these have been genotyped on a variety of different platforms, we use the LD structure that I mentioned earlier with respect to a marker SNP being correlated to many other SNPs in order to impute the data and actually we've achieved about 2.7 million SNPs that we have looked at throughout all of these cases and controls. And this was done using mock and mini-mock. For the dietary variables, most of this data is collected using questionnaires, which differ across the study. Harmonizing this data was done in a centralized fashion at the Fred Hutchinson Cancer Research Center. For the data that I'll show you today, I will be presenting the dietary data as sex study-specific quartiles, coded as 1, 2, 3, 4. So our overall main effects for red and processed meat across the studies, and this is about, we looked at, it's about 10,000 cases and 10,000 controls, was in fact very, very significant. This is the per quartile effect for red meat with an odds ratio of 1.15, and it was very similar for processed meat as well. And there was no observed heterogeneity across studies. For fruits, vegetable, and fiber, we observed a protective effect that was quite similar across fruits, vegetable, and fiber, with about an odds ratio of 0.9. For folate, folate was looked at in terms of total folate, so this combined the both dietary forms of folate as well as supplements. And again, we did observe, as had many previous observational studies, a protective effect of total dietary and a slight increased risk for the supplements, which, as some people have thought, that folic acid may be different than dietary folate. So the statistical analysis has been an area of extreme discussion in G/E. I'm going to just focus on talking about mostly the using a logistic regression approach. These are modeled using the main effects for the diet, gene, and the interaction effects between the G and E. There are other methods we've also looked at. This particular method was called the cocktail method, which essentially is a more efficient statistical method in order to decrease the number of multiple testing. The data I'll show you here is just a fixed effects meta-analysis. And so for our results for processed meat, here's just a Manhattan plot of the chromosomes and the p-values. We identified one very statistically significant hit. The SNP is called RS 1413094. It's located on chromosome 10. We didn't see any deviation from the null, and so we correctly adjusted for population stratification. This particular genetic change, when we looked at it in a locus zoom plot, we could see that there were a number of other correlated SNPs in the region that showed a high significant interaction with processed meat. And it was located near a gene called GATA3. This particular genetic change, these are the three top hits. They're all nearby the antisense gene or the gene itself. It has a high frequency. This means that about 1 in 3 people carry at least one copy of this particular gene, making it quite common in the population. When we looked at across the studies to see if there were any differences, we didn't observe any heterogeneity. And so in layman's terms, when we look to see the dose effect of having different genotypes and different levels of processed meat, individuals that did not carry the allele did not have a very significant, it only had a 3% increased risk per quartile associated with processed meat. But as you can see here, individuals that carry 2 copies of this particular genetic change had a 39% increased risk. So the difference, what it means is that individuals with this particular change have an even higher adverse effect from consuming processed meat. What's interesting, I won't go into detail about this particular analysis, but this particular genetic change is located 1,000 base pairs away from this gene called GATA3, but there are also a number of other genetic changes that are in high LD. What we know about this particular gene is that it's very important in immune response. So we do not have because this is an agnostic search, we don't know if it's the causal allele or what the mechanism is behind it. But some of the hypotheses that have come out have been related to whether it was if it's an immune response, if the gut microbiome could be quite important in this to explain this result. So for vegetable intake, we did find one interaction, and that interaction is with a particular genetic change on RS2450114. This was actually previously published in another publication as well, identifying this interaction. And with folate and folic acid, we've only begun to look at pathway-based approaches. This is the folate diagram. Probably the most consistent result for us has come from one particular gene. This is a functional SNP. So unlike the GATA3 mutation that we are not quite sure of, this happens to be a particular change in MTHFR 677T that we actually do know how it functions with respect to markers, biomarkers, including methylation and nucleotide synthesis. From our genome-wide association analysis, genome-wide using over 10,000 cases and 10,000 control. As you can see here, we did not see a significant effect for that, and we're looking into it a bit more. So I just want to end with some comments about what this all means in terms of prevention. There are at least two really important aspects to looking at genome-wide interactions with diet and with other types of genetic changes, and that is whether or not we can change behaviors in patients and families or in the community. So people can actually, right now, can be genotyped. I'm going to show you the results of what happened after we've published this work, but people conceivably, if they know their genotypes and these are confirmed interactions, they may choose to change their dietary habits, increase their physical activity, stop smoking, and other decrease obesity. Unlike our genes, we cannot change our genes, but we can certainly change these types of behaviors. And the second biggest area in this work is risk stratification. We heard yesterday about the heavy cost of doing colonoscopy in Europe and throughout the world. We certainly know right now that not all people need to be screened, perhaps, but we haven't fully understood how to stratify people into high-risk groups and low-risk groups, and some of this work may help in those types of efforts. So what happened when we published this work is it went throughout Twitter. The journal put out a message, and so I got an email. I got a Twitter response from one of my viewers telling me that they were actually TT genotype and asking for some recommendations. But apparently we found out that it is on the 23andMe panel, and so I think this is an example of how some of our work in this area could in fact change behaviors. In terms of risk stratification, this is work done by Dunlop. This is just showing just 10 SNPs, but you can certainly see here from his analysis that cases tended to have far more adverse genotypes than the controls, and I hope this shows up properly. When he looked at the absolute 10-year risk for colonoscopy, what the estimations is that, so this is just the population-based risk, but we know now that having different alleles may actually increase that. So in this case, with the highest risk for having more than 14 alleles and being family history positive. So this is work that essentially demonstrates that perhaps some of these GWAS studies can in fact differentiate individuals at the highest risk from those in the lowest risk, and that colonoscopy can be far more tailored. So in summary, gene-environment interactions have been very useful in two areas. One is in discovery, in etiology, and some of this work definitely needs to be looked at in terms of post-genome functional work. This work cannot be done without huge consortiums and that facilitate large sample sizes. Of course, interpreting some of the work right now without doing the functional work should be taken with caution, and we need to continue to study that. But this work has a lot of important implications in the future for prevention. So I just wanted to acknowledge the studies here and the number of collaborators, some of which are here, that have contributed data to these types of projects. Thank you.
So I'm afraid we are running a little bit late. Is there one burning question? Okay.
How can you be sure that persons are telling you correctly what they are eating? I can give you an example. When we ask for alcohol in Denmark, men tells you 21 units and women 14 units. And why that? Because that's recommended by the health authorities. When we ask today, they will still say men 21 and women 14. They have forgotten or they haven't heard that the recommendation has been lowered to 14 and 7. So I think there are problems in such studies.
Absolutely.
Not a complete big number.
No. Actually, that is, it's always been a criticism in any kind of nutritional epidemiology is, and all of these data that I've showed you today are actually done on risk factor data and questionnaires. So it's not dietary recall, diaries, and much more detailed information. But the issue here is that if you want to study the genome and how our genetic susceptibility may alter what we eat, you need huge sample sizes. And you cannot achieve that with just doing very small focused studies that collect a lot of data. So it becomes an issue. So how I would see it is that these are big agnostic studies that may point to things that then we need to follow up and do more detailed work in different populations where we can collect the data, as you said, because right now these are just, and even the definitions that I was using for red and processed meat, other people would argue are too broad.